sleep in their dens.
dens. Hello.
Okay. Hello.
Good morning.
Good morning.
Welcome.
Welcome.
We have an anniversary day today, as it says in the fun fact.
It's our anniversary. It's our remote anniversary.
It's our this room anniversary.
This room anniversary.
When it was taken over by the University of Guelph.
I don't even want to watch the recordings originally.
I went back and I grabbed something.
I
Hold
saw
on,
that.
I
Wait. Though
yeah,
we have to screen grab this because this is beautiful.
Got it.
Yeah. Okay. Cool. But before that.
Oh yeah. Okay.
Today is Remembrance Day, and it's a super important day, and a day, to just reflect on everything, even the stuff that's happening now, on service that people give to their communities without any concept of benefiting directly from that service. I think is just an incredible thing. And I think that we're seeing a lot of that happening right now, too, with people giving of themselves for their communities without expecting to see the benefit of it within their lifetime, or even to give up some of that lifetime. So
we were talking to family last night where our nephews were donating. They bought their puppies online. Maybe you did also from the Legion, and they were where you can, if you did that, you saw that you can make the donation in for a particular person. So they were asking about who in the family had which of our grandparents, great-grandparents, great-uncles, those kinds of things. In my side of the family had, and in your side you had an uncle who landed on
D-Day.
On D-Day.
On beaches in Normandy. He was my uncle, so actually one generation younger than most of us. So usually it's our grandparents. But this was my uncle, and the reason why is because he was really old. So he passed away recently. He was 92 years old. He was born, so if you do the math, all the way back into the 30s. And he landed in one of those ships, on one of those boats that you can still see on the beaches, and 90% of the people that did that were killed instantly. My uncle made it. And experienced all of that, and I had the benefit of knowing him and hearing his story.
And I'm sure lots of families, maybe you know about them, maybe you don't know about them. In my family, my grandfathers, neither of them were allowed to serve, and they both tried.
Yeah, mine signed up young, so he was 16 or 17.
But their jobs were coded as such that they weren't allowed, and so they one of my grandparents, my grandpa Bazinet, kept going from province to province, and he thought if he crossed provincial borders he could could sneak his way in. But they were like, no, you have to stay, and it bugged him for the rest of his life. And I have great uncles who are buried in Egypt and Palestine from the First World War.
Yeah. Sorry, he was born in the 20s. He was very old.
He was very young.
He was very young for that.
He was very old to be your uncle.
He was very old for my uncle. Very young. He was always an old man. But anyway, so
somebody here has been to the beaches in Normandy.
Yeah.
I haven't. I've been near, and it'd be over. There'd be lots of Smith.
He went back. He went back to those beaches where he landed, and they figured out exactly which beach based on the records, and that was a day.
Okay.
That was a day. Okay, so
today
our remembrance of this incredibly significant moment. You kind of set moments in your life, right? Like sort of if you have like a significant moment, like when you get a new puppy, or when
January 3rd.
You have a child, or when
January 3rd.
Smith thinks you actually start dating someone, but it wasn't until much later, not January 3rd.
7 p.m. January 3rd.
Anyway, you kind of mark these moments, and things happen like before or after. And for us, before or after is March 13th, 2020. That's like a new milestone. And this lecture is actually the first lecture that we delivered remotely. And so we'll probably tell you stories about everything that went wrong on that day.
Is that slide still in there?
Yeah, the slide is still there. I'm just trying to clear all, but it's so beautiful. I don't want to clear, but we've screenshotted it.
Okay, yeah.
So we're good. And moving on. Yay! We started out with WebEx, and that was a monster mistake.
It was so bad.
It wasn't our mistake.
No. In fact, we had prepared. We were ready.
Oh, yeah.
We saw this coming in January.
Yeah.
We wrote a proposal about which programs we needed to do to make a digital transition should this come down the line.
Because we're uber-keeners.
We were tapped on the head and be like, we've got this.
Yeah,
we've got this.
don't worry.
Don't think above your pay grade.
Yeah. We'll tell you what tools you can use.
And then the tools they gave us,
WebEx.
we couldn't mute all the mics. It was bananas. It was like we heard many parents
so many people
in the background going, "Are you listening? Why aren't you listening? What are they talking about?" And it's like, we can hear all of your parents all at the same time.
It was a disaster.
Yeah.
Anyway, so we started making requests for Zoom in January, February, March. We got the Zoom account the day before this semester started.
Yeah.
So
then we bought it ourselves.
Institutional proactivity.
Yeah. Anyhow, now we have 10 minutes left.
Yeah. Let's start class.
The frogs. I have a particular affiliation for amphibians. In a previous life prior to, and actually this life as well, because Amanda is now working in our labs on amphibians. She's really working on plants, but she's working on plants that kill amphibians,
that eat them.
Kind of a cool story.
Which is awesome. Yeah. And so Day of the Triffids.
Yeah, is it this lecture or the next lecture that we talk
later?
About? Okay.
Zip. Today we're going to talk about fish.
Oh, delicious fish.
I don't know why then we have a frog, but it's a really cool frog. So there we go.
My great aunt was born in a bomb shelter during
36
days of bombing in Malta. Wow.
Oh, wow.
That's quite a story.
That's incredible. Thank you for sharing that. Wow. Okay.
J. Beavis.
Okay, homework. Here you go.
Oh, yeah. Foxes.
We'll stop
sleep
talking.
in the
Stop
Arctic.
talking. Okay, so we asked you to match the different levels and the time scales associated with each of the statements.
That's just
is that some? Oh, mute your mic. Okay, so
hold on. I just had to mute all to do that, and now we will unmute.
And now I'm muted.
Yeah.
Hello. I can't.
There it is. Yeah, there you go.
I can't.
Oh.
I've tried several times.
I'm sorry.
Yeah. Thank you. Okay.
It's just in honor of WebEx.
So let me throw this at you. So the idea is, and I think I mixed up, look for the correct match of the time level and the biological level of organization. So take a look at this. Here it is, both in the polling and on the slide. I have a question.
For me?
Yeah.
What's the question?
Do you have the poll? Did it fire up at you?
Mm-hmm.
When you click...
I can't. As a host, I don't think.
Oh.
I have no I can point the screen aggressively, but then I just get a greasy, sweaty smear on my screen.
When you click, can you change your answer?
Let's ask. Zoom wants to fuck off.
Shh.
Did it catch it?
No.
Good. What did they say in Battlestar? Frack.
Frack.
Frack off. Nice. Frackety frack.
Okay.
What's the question for them?
When you answer
Frack off.
a Zoom poll question, can you change your answer? Maybe somebody has answered. They've put no on the screen, but maybe that just means no, not that one. No, you cannot. Thank you. I got the answer.
I'm gonna fucking break this.
Yeah. Okay.
Is that Dawn?
No. Okay, most of you have answered, and I haven't seen that many come in. The participation is a little bit lower than normal, though. So I don't know if it's just a more difficult question or you're choosing not to answer, but let's just give you a few more seconds. Okay. The vast majority of you are getting it correct. End polling. Share results. And the vast majority of you got it correct. So it is this one here matching appropriately the statement. So generational and organism level for sure. Are there any that like you were confused about and you ended up guessing the correct one, but you'd like us to tell you which one is which? Just kind of highlighted on the slide. I'm happy to quickly go through that. B, okay. Fur thickness. Here, I will stop sharing the results and put that over here. Good. Okay, so fur thickness of Arctic foxes increases in autumn. So fur thickness, it would be okay to say that that's an organ. So that's not bad. It's the acute thing that's the problem. Because something that increases like over a season or, you know, in order to prepare for a seasonal exposure from the perspective of the fox, that would be chronic. Arctic fox body fat content is lowest in July and highest in November. So that also, because it changes seasonally, that would be chronic. The adaptation of being able to do that is indeed evolutionary. But the idea is, does the state of that trait change or fluctuate? And if the answer is yes, then it's either acute or chronic. And if it does so over these seasonal or longer periods of time, then that would be chronic. The fat content, I would say that that's at least at the tissue level, but I could understand why you might say molecular. So, you know, that's not necessarily wrong, but it's really about the generational, which is totally not well matched. Arctic foxes reduce metabolic rate to save energy through the winter. When prey is scarce, again, that's a chronic exposure or chronic response because we're talking about something that's seasonal. And reduce metabolic rate. So if we're talking about metabolic rate, we're talking about like the system. So I would put that as an organ system. Again, there may be some molecular arguments for that, some molecular cues that fluctuate, but again, it's really about the time scale that's mismatched here. Okay. Some people want the first one. An Arctic fox curls into a ball to reduce exposure to the ambient temperature. To me, but also that is acute, not chronic, because that's curling into a ball for a short period of time. It's not gonna, the equivalent over chronic would be like to hibernate, right? You curl into a ball for the entire season. That's not this. This is just shorter periods of time. It is definitely at the organism level though. Okay, I hope that's okay. We can clear. There we go. Okay, this is what we're going to do today. We're just going to play around. We're going to talk a little bit about scientific method, get you to kind of engage in that. And it's kind of timely for the midterm. So we want you to kind of really think about hypotheses and think about how you might test. So we're going to kind of massage your brain into thinking about those types of things.
And when we talk about ectotherms today, we're talking almost exclusively about fish.
Fish.
Not the diverse ectotherms.
Oh, Smith is so sad. We're going to talk about fish and why they don't freeze. So this is kind of a super fun physiological conundrum. You have a fish in water.
I feel like there should be a meme.
Yes.
Why don't fish freeze?
Why don't fish freeze? Okay, so somebody make a meme, please, and we will share it and laugh. Okay, fish. So the problem with fish is this. Fish are freshwater, right? We are freshwater. 70% of us, lots and lots of fish is freshwater. Freshwater freezes at, actually it starts to freeze at 4 degrees Celsius, but it freezes at 0 degrees Celsius. And that's a super fun physics problem. Marine water, like salt water, doesn't actually freeze until about minus 1.8 degrees Celsius. So fish that live in cold marine systems are basically able to freeze at 0 degrees or starting to freeze, but their water is much colder, and so why don't they just freeze? Okay, does that make sense? So there's like some stuff, because we know that fish can live in really cold water, so clearly they're not frozen. So what is it about them that allows them to be largely freshwater? It is possible that they're just saltwater, like their internal organs are all saltwater, which would be amazing and is not true.
What kind of a regulator would it make them?
Oh, that would make them a non-regulator. That would make them a conformer, an ionic conformer, right? Yo, let's put that on the final exam. Okay, yeah. So, if we want to kind of study this and figure this out, we go through a process, right? So we've observed the pattern, okay? Why don't fish freeze in cold salt water, okay? And now we start to develop hypotheses and we make predictions and we start to ask how we might be able to test these predictions. And how you might then interpret the data. And so what we're going to do now is kind of walk you through a story, and then we're going to like slow it down and allow you to kind of play or intervene within a different story, if that's okay. So the observation of the pattern is that there are no frozen fish, at least in the marine environment, in their natural environment.
Captain Highliner.
Yeah.
Excluded.
And some initial observations were done by people a while back. This is like a classic story, right? So it's not
There's some buzz about it in the chat.
There is? Okay, yeah. Good. Okay.
In fact, there's a Wikipedia entry already.
Is there really?
Yeah.
Amazing. Okay, so here are some initial studies that were done. So this is like 1989. This is old school, right?
1989.
Do you remember 1989?
Oh my God. I had a mustache.
Oh no.
I had a mullet. I was like...
You were like super cool Valley kid?
Yep.
Okay.
Whatever had happened in the rest of the world in 1987, it had got to the valley by 1989.
Amazing. Okay, so what we'd like you to do is to take a look at these data. So these were like a preliminary study, first observations into the physiology of what was going on in the bodies of the fish. And we're going to ask you to go to menti.com, and I'll just launch the poll right now so that you can start to participate. What we want you to do is spend a little bit of time looking at these data and come up with like 2 conclusions, okay, and include them in the same little text box. 2 if you can. If you can only come up with 1, no worries. See if you can come up with some conclusions about that we can make from these data. They're working. It says voting is closed. What does that mean?
About what?
Menti, what does that mean? Voting is closed. Can you try?
'cause I can't get online.
You can't get online?
Are we still online?
You know, it's hard to tell.
Sometimes it's hard to tell.
Yeah. Is anybody having trouble getting into Menti?
Yes. I have to wait for the presenter to open it, it says.
That's weird. I did. Okay, we'll just do it again. No problem. I'll reload and restart. Yeah.
I can't, this is a new thing.
Yeah?
Yep.
It says voting is closed. Okay, Menti is not
Okay.
working. That's weird.
And
Okay, please go over to the chat. Sorry about this. I'm gonna...
it says it works now.
Now it works?
Somebody
Okay,
just said it works now.
works now, it
But we're
says.
not. It's working now.
I have no idea why. Okay, woo. Okay, so Menti is working again. Please feel free to share there. If you don't mind doing it not really anonymously, you can go into the chat as well if you prefer. But this is just Menti is a way of making it super anonymous, so you can say something ridiculous and we won't know that it's you. Okay, thank you. We're starting to see shares. Okay, this is really good. Here we go. As soon as we get like quite a few, I'll put it up on the screen so that everybody can see. It's the ghost of the first remote lecture. Yeah. Trauma. Like I'm still feeling a little bit of like, stress.
You didn't even break out the last part of the story.
What was the last part of the story?
Well, the reason that we got it, beyond the fact that we just bought it ourselves,
yeah.
was our discussion of it publicly on Twitter.
Yeah, sadly, that's what it took. Yeah. Okay. Here we go. Here's what you're sharing, and I'm grateful. Okay, so antifreeze. Yes, that's a thing. Antifreeze turns fish into Elsa. Cold doesn't bother me anyway. There we go.
Let it go!
Okay, time of year. That's a thing. Yes. Low plasma freezing point corresponds with high antifreeze protein concentration. Very good. So there's a direct correlation there. Antifreeze protein concentrations is highest in the summer months. Plasma freezing point is lowest in the summer. Very good. Freezing point is highest at the beginning of the year. Super. Protein increases during cold months. Very good. And yeah, these are good. And some of them are delightfully funny, so that's fine. Flounder are delicious. That is a very accurate conclusion. Not associated with these data, but that's okay. More antifreeze protein concentration equals lower plasma freezing point. Less antifreeze protein equals higher. Yes, yes. I wouldn't count that as two statements though, because they're just different direction, but yeah. That's good. Okay, the freezing point goes hand in hand with the protein concentration. It sure does. Fish only produce antifreeze proteins during colder months. Very good. Okay, excellent.
Does this have to do in the chat? Would this have to do with set points throughout the year?
Potentially.
Yep, potentially.
Yes, potentially. Now, set points though.
And good retrieval of last lecture. In this
Yeah.
lecture, so.
Super good question. So
Yep.
set points are maybe on a shorter time scale. Like you wouldn't actually see in this fish's profile if things were measured like month to month. You wouldn't necessarily see adjustment of set points over because that's like kind of a day-to-day thing. Like it doesn't take very long to adjust your set point usually. So it depends on like the sampling frequency, right? So you'd have it day to day, and then I don't know. It's too early in the morning for this.
No, I reject that one because you've used the correct two. There's like a whole bunch of other twos you could have used.
It's true.
And you've used the correct two. So two.
Yeah. So your brain's working. Okay, cool. Thanks very much, everybody. That was great. Now, and some of them were slightly off, and so, but we want to kind of keep it. So just kind of keep in the back of your mind. Some of them were like really good. Some of them were just like repeating basically the opposite statement. And to me, that's not two kind of conclusions. That's just one with different directions. But anyway, you're doing really good. Some of you linked the two things together. Some of you looked at them independently. That's also fine. Super, super good. Okay. So antifreeze proteins. So now we have a new set of observations, right? So now we go back to the initial thing. It's not that fish don't freeze. It's that antifreeze proteins increase in the fall and decrease in the spring in marine fishes. That's the observation now, right? That's like the next step, the next thing that we do. The question might be, what's the trigger for this? Like, what causes this? What is the sort of the stressor or the stimulus that causes this particular thing to go to start upregulating antifreeze proteins production, right? In cold water, marine fish, so the hypothesis, the trigger for synthesis of antifreeze proteins is, what do we think? And then, if the hypothesis is true, then what would we expect to see? So that's what we're going to work on right now, okay? Looking at some of the things that you've said, let's take a look here. Here we go. Look, the plasma freezing point in the fish decrease in temperature during the colder winter months. Okay, cool. Antifreeze proteins, here we go. Minus 1.4 is the sweet spot. That's a really good conclusion. I love that. Yeah. And other things, lowers, I'm looking for a mechanism, like for a hypothesis here during the cold months. Yeah, cold doesn't bother me anyway. Okay.
People have put in the chat.
Cold?
Temperature.
Temperature, great. So let's do temperature, okay.
And some directional ones that it's definitely not another thing because there's no way that other thing can get underwater.
Right, okay, very good. So, temperature. Let's think about temperature. What could we do next with, say we had a bunch of fish, right? What methods could we actually do in order to get at this, right? To figure out whether or not it is temperature and whether the minus 1.4 is the sweet spot and all of that. So let's explore that again, hoping Menti actually works.
Same code?
Yeah, it should be the same code, but now I have to like refresh it. So start thinking about methods. What could you actually do? Like, you have all sorts of money, all sorts of resources. How could we figure out whether or not it is temperature? It should be working. Go ahead. Share on the chat or on Menti.
No, I'm not even seeing the things that I have posted in the chat. I've just been kicked off.
Yes, do not worry about ethics at this point. So, yeah, we are like evil scientists, if you want. Okay.
I don't think I'm online.
No, you're not. Oh, okay. Oh, just, yeah. Or this one. Smith is offline, so the chat will not be personed for a few minutes. Okay.
You should have seen the thing he's been uploading.
You're all quite delightful. I'm enjoying these shares. Okay, so here we go. Start freezing live fish. Not the most ethical, but would work. Yes. Ask the fishes nicely. Super. I love it. Okay, expose to different temperatures over periods of time. Very good. Put fish in different water temperatures and see what happens. Put fish in cold water during the summer months and see what happens. Nice. I love that. Compare the antifreeze blood concentration of fish in a tank of gradually decreasing temperature to a stable temperature tank. Super! You've got a control here, right? A stable temperature tank. Okay, but then, question. 2 tanks only? How many fish in each of them? Is it better to have like 30 tanks, half of them are changing temperature, half of them are not? And then is it 30 tanks with 1 fish, or is it 30 tanks with 10 fish each? What are some of the problems associated with each of these designs? How do you know that you've sampled enough fish? What happens if you have like a power failure and your fish die? All sorts of questions, right? Control and treatment groups, laboratory with multiple treatment variables. That's very important. Do fish in warmer climates behave? Oh my God, I love that.
Yeah.
That's super good. So that's like ecological experiment, right? So amazing. Well done.
So you're going to move your fish around like you moved around your plants along the
Yeah. Same water concentrations and volumes. Use 3 tanks each with 20 fish. Nice. Change temperature of 3 tanks and see what occurs. Very good. Okay. Get a bunch of swimming pools and have different temperature water. Have a few thousand fish chilling in your backyard in different pools. That would be amazing.
Yeah, that's somebody's thesis.
Yeah. Internal thermometer to track the fish. Nice. Cold water. Yeah, because maybe the temperature doesn't change inside the fish, right? Like there's that. Super. Eat the fish. Yes. Okay. Wonderful. Good. Water heaters. Nice. Have a controlled plot and then change temperature. Yes. Okay. Really good. So all of these are things you could do. And the question then becomes, if you only do one of them, have you answered your question? There are flaws associated with every design. There is no perfect design. At least, within the fairly limited resources that you get to do science these days. And so it's really about understanding what the pros and the cons are of each of your designs, right? Do you have 30 tanks each with 1 fish, or do you put 10 fish in? If you can't afford 30 tanks, then you've got to have 15. Is that okay? Or should you just give up science, right? And most of the time, the answer is it's not ideal, but it will tell us something. It will inform some part of our original hypothesis.
So if you go back as you're checking the slides later on and you look at the 8th slide where we talked about the scientific method, this is discussing that 5th point where so you've followed this pattern through, and how does your confidence in your hypothesis, in your proposed mechanism, change as a result of what you've seen?
Yeah.
And so sometimes it will have changed it dramatically in the ideal sense. When you're writing out your master's and you're writing your proposal for your PhD, you will delineate like the perfect thing that will rule out all other things. And then life will happen and you'll do your experiment and you'll realize as you're doing it that there's other things that you might not have controlled it for as well as you wanted to.
Can I share a story about like a personal science story about you?
I think they will
Okay, so
change the answer.
Smith does work, right, in Costa Rica and spends a lot of time marching up volcanoes, right? And then as you do, and like measures ants and does things along the way up the volcano. And
as you do.
Yeah, and it's a big volcano and there are a lot of ants. And so that's pretty much occupying 100% of Smith's time when Smith is doing field work. And so then Smith writes up some stuff, makes a discovery, writes up some stuff, submits it to the journal in order to get reviewed by peers. And the peers say, well, this is a flawed design because there's only one volcano. Now.
Smith can't report what he says when he reads this review because
it's funny. So yes.
Insert GIF of table.
Yes, there is only one volcano.
And there's only one Earth.
And so there are challenges. There is nothing wrong with Smith's design because Smith has multiple tracts going up the volcano around at different exposures, has controlled or has sort of mitigated the problem associated with just one volcano. And so, because Smith is a smart person.
And Smith has worked on, and this is the case, working on different volcanoes doesn't change that because mountains, one mountain does not equal another mountain.
That's right.
All of the things are different. The age of it, the exposure, an elevation at one mountain that is large is not the same elevation as another. And,
yeah. Okay. So this is fun, and this
yes,
also
and this comes up all the time. And this is not this is recent. This story you're recounting is very recent.
Yeah, and because it happened in our kitchen recently. But so this also
Hulk smash.
Some of you were interested in biological philosophy. this is another aspect of biological philosophy, scientific methodology, pseudo-replication and all sorts of really cool things require philosophers to think about them.
And it's important because so there's some people talking about I would just give up, this must be frustrating. And yes, it is frustrating. So when I say Hulk smash, like I was very frustrated, and you don't, as you go on and become these kinds of people, to do these jobs that we're doing, if you replace us, you don't write the response to those reviews on the same day, because your first response is often an emotional one. Yeah. How could you possibly not have understood what I'm thinking in my head?
Yeah. And here's a pitch. If in a few years you are still interested in biological philosophy and you would like to think about doing a master's degree or some research, I would love to get into looking at science methodology from a social justice perspective, from a equity and diversity and inclusions perspective. How do we do science in ways that are exclusive, and how can we make them more inclusive? So I'm just throwing it out there. Okay, good. Yep. Now, moving on.
Wasn't done.
We're not done.
No. So.
Well, but okay,
we have but
15 minutes.
You just made a plug.
I totally, you want to, okay. Yeah. Yeah, Smith needs students to go up mountains.
One of the, no, this is no plug. This is just the end of my sentence, which was that those frustrations come because people are trained in science. We're trained historically in different silos. And physiologists are trained to do things in units of 10 and to control all of the other things. And I'm trained to have dirty boots and to be covered in the rain and to do the best that you can in the world that exists. And sometimes those two philosophies, those two trainings intersect, and they should. And you get really valuable conclusions from that. And so sometimes that frustration, the initial one, means that you have to mold what you've been trying to do into another way of thinking, or at least make it give it a Rosetta Stone, give it a translation to say, yeah, sure, yes, I'd love a whole set of identical volcanoes. However, this is the world.
Yeah, very good. Thank you. Okay. Thank you for letting me.
That's a neat project.
Do you like it?
I do. Yeah.
Cool.
Yeah.
You want to co-supervise it?
Sure.
Yeah. Yay.
Bring it on.
You can get a white man's perspective. Wonderful.
Yes, probably everybody's waiting to think, what does the old white man think about this?
Amazing. So that would be really cool. Let's do that. Okay. So.
Why would you invite the old white man?
Because we need to have a historical context. Okay. So. What are you going to do, right? And many of you totally hit on completely reasonable things to do next. Here's what a group of scientists did. So we're going to tell you a story, right, of what they did. So they had a control group of 20 fish, and they had them at 10 degrees Celsius. They had an experimental group of 20 fish, and they decreased the temperature to minus 1.5 degrees Celsius, so below freshwater freezing point. They measured the plasma or the blood antifreeze protein concentration, and they expected some results, right? So imagine, they lowered the temperature over time. They measured these things over time. And here's what a blank graph would look like, right? So we have time on the x-axis, we have antifreeze concentration on the y-axis. So high antifreeze concentration up here, low antifreeze concentration over here. Why don't you go ahead and draw a line that you would predict would be noticed, that we would measure if temperature was the trigger. So we have a blue line, if you can, for the control fish, and a red line for the experimental fish. See if you can do that. Okay, so control, good, you totally can. Yay! Good, control fish, nothing. Red, experimental fish, excellent. Now we know we can do that. Good. Thank you. Amazing. Yes. Okay, so visualize that in your head, and I love to do this. This is so important. Oh, that's interesting. I like the bell curve. This is a fundamentally crucial part of my process of science. Smith does it too, Because if we draw these graphs out before we actually go and measure, it becomes so much easier to figure out what's going on when we actually measure and draw the real graph. So now we have our prediction.
It's kind
right?
of it's a touchstone. It's literally a touchstone that I encourage students to draw in their field notebooks or in their lab notebooks to be like, what am I doing? What am I expecting to see? Oh, it's agreeing, not
it's
agreeing.
So not that,
whatever.
right? Okay, so
Pictures.
Let's clear all drawings and I'll show you what they actually measured. What do you think? So this is where imposter syndromes kicks in for a scientist, where you go, yeah, Ella, yes, I'm terrible. I don't know what I'm talking about. What the fuck is happening with my data, right? Yeah. And it's a low moment and it happens to everyone, right?
What the frack?
What the frack? Very good. Okay, so this is what happened. And now you go back. The fish broke. Yeah, maybe it was your methodology. Maybe it was your experimental design. Maybe it was your Excel spreadsheet for the data and it just didn't show. So you start to go back and you check, right? And what should we do next? So let's think a little bit about what we might do next. Let's say that there was nothing wrong with the experimental design, that there was nothing wrong with like any of the mechanics of the experiment, and that these are true data. What would you do next? We might start to look for another variable. So if you take revenge on the fish, eat them. Try measuring. I can't read variable other than temperature.
Yeah,
very good.
In the chat, they're saying, did they decrease the temperature over time or just plot the fish? So
yes.
Methodological concerns, temperature perhaps isn't what changes, I guess.
Yes, exactly. Okay, super good. So maybe it's not temperature.
Maybe that's the result.
If you take a look at the original observation, so if we go all the way back to here, you can see that what they've got on their x-axis isn't temperature. It's month, it's time of year. And that can be a proxy for many things. So what we've done is we've made an assumption about what that proxy is when we say it's temperature. But it could also be something called photoperiod.
Yeah.
Okay? Photoperiod is day length. We're all experiencing shorter day lengths right now, which totally sucks for my mood. And so we know that the day length or the amount of light throughout a day changes. And so what they did was they reran the experiment, but this time with different day lengths to say, is it photoperiod that's the trigger? Okay? So imagine again along the x-axis, but this time instead of over time, imagine the different photoperiods, right? So we have, for the first one, we have 20 fish with 10 hours, 20 fish with 8 hours, 20 fish with 6 hours daylight. Now it's going to be a bar graph this time. With the y-axis is going to be antifreeze protein concentration. So in your head, imagine, if you can, what you would see. Make that graph in your head. It's a bar chart, like a histogram, with 3 bars, one for the 10-hour fish, one for the 8-hour fish, one for the 6-hour fish.
It's a Goldilocks graph, right? You've
right.
got lots of light, you've got a little bit of light, and you've got not so much light.
Not so much light. And then you have a control group with 12 and 12 up here.
Yeah.
Imagine it. Have you got it?
I've got it.
You've got it? Okay. What are you imagining?
I'm imagining more with less.
More antifreeze proteins with less light, right?
If this hypothesis is supported.
Good. Here we go. Is it supported? Do you see it? Is it a thing? Ella, are you happy? Ella's happy. Yes. Good. Yes, Ella's happy. Very good. Thank you, Madeline. That's perfect. Yes, exactly.
Now, somebody's drawn a line there, and that's that's good for visualizing what's happening. Why wouldn't these people have done a line?
Ooh, that's a good question. Do you want to just slow down on this stuff and skip
I
the
was
other
just
stuff?
I think we should push
Okay.
foxes till
Yeah.
fur till tomorrow.
We'll talk about
and by
yeah.
tomorrow I mean Monday.
Okay, yeah, so we can end on time, and we'll just add all of that stuff later. Okay.
Because they're comparing values instead of connecting them. Right. There's no intermediate
Very
values.
good.
If they could have set it up like that, but maybe because of resources, maybe because of the number of volunteers the graduate student had working and the number of pools that they had.
Yeah.
They're separate things.
Yeah, super
And
good.
that's important to recognize. So it's a nice thing to emphasize and to tell your story, and you might actually do that as you're standing there presenting this at the conference on fishy proteins.
Discrete, Caitlin, yes.
Nice.
Exactly. Possible variation of different plots, so no set values.
They're
That's
so
interesting.
discrete.
They're so discrete. Super good. Yeah.
Okay.
We're gonna spend a little bit more time like poking at this methodology, and we'll skip to the end. And we won't assign homework because we want you to really think about methodology. This is a monster Easter egg for what's coming up on your midterm, is to think about methods, to think about hypotheses and how you might test them. It's part of the EPP as well, right? So it's all like part of the same type of theme. But really thinking about, how we would test something and how it might contribute to giving us an answer. This one graph, though, is that it? Do you just pack up your scientific career and just say, I know the answer, I'm moving on?
Drop the mic.
Yeah.
So I like what somebody sketched in on top of the lowest category of the control.
Here?
Which is them trying to estimate the variability within each one of those treatments. Because right now what we've shown you is kind of a simple story. It's a cartoon of the actual study, which is like there's a block.
Yeah,
But
what
about the variation? What if we had, that's good, do this. I like this.
What if we had this?
Oh, you've made two things. Wow.
I've made two things.
Yeah.
Have I complicated your life?
You've enriched it.
Ha! Okay, let me clear because I want
wanna say that.
to. I want to be able to. Oh, sorry, shit. Let me just redo
No,
that.
I got it.
You got it? No,
Yeah,
it's okay.
I believe I've got it.
What if we had this, and then this, and then this? Okay. How would that change? How would they control the how would they control the photoperiod? That's an excellent question. You just turn the lights on and off, and you have separate rooms, and you can go to Aqualab and see this type of stuff, on Guelph campus when the world opens up again. They have separate small rooms where you set timers, and you have different day lengths, right?
And you play a little game of daytime, nighttime.
Daytime, nighttime.
Daytime.
Nighttime, daytime. Okay. So somebody has added these standard deviations. Probably they could be standard error, but they would be smaller usually if it was standard error. But what happens if we add this? Does this convince you that photoperiod is the trigger? Yeah, more variable when daylight is less. Super good. Are you going to conclude? And the thing that's really challenging about a graph that has bars like this. Did somebody write something funny?
Yeah, Alan.
Steve. Okay.
It's not somebody suggesting not as convincing. That there might be a flaw in the experiment, or that fish respond quite differently once they are exposed to dark for long enough. More variability with shorter photoperiod. Control photoperiod by exporing fish to lamps. Error is a lot. Error bars overlap, so not the greatest set of data.
Yeah.
See if it's the same in other environments.
I love how can it affect deep underwater? Yeah. Super good. You're thinking. Yeah. Okay, so yes, these types of things, mean that you've got an entire career in studying photoperiod as a trigger for physiological mechanisms. And that's the good news, right? You don't need to have these solid things that, it doesn't have Mendel's data, right? The P guy. Turns out that they were a little too perfect, and no one's been able to reproduce the perfection of his data. And so there is, somebody was p hacking a little bit, right? Anyway, so if you put this through a statistical analysis and you look for your p-value, that's why he laughed.
Because you see P's
and Mendel and P's. We don't have to explain it, or else it's not funny anymore.
It gets funnier when you explain a joke.
So if you look at these data and you do like a statistical analysis, and you're looking for a p-value of less than 0.05, you're not going to get it if the error bars overlap. And so it's a stressor. But I say this, and this is kind of still like a new age scientist thing. Who cares about stats? We're looking at you just like, right? That bot, yeah, yeah. So a good figure can give you a lot of information about the direction of the data. Whether or not they're statistically significant can also help to inform whether you need to refine your methods, take a look, and kind of maybe explore other alternatives. There's nothing wrong with statistics, but often statistics put those blinders on us in a way that we can't appreciate the full biological context.
And I think the other thing to remember with statistics
Am I wrong?
is, I know I'm about to agree with you,
okay.
And in a different way.
Okay.
I think the other thing to remember with statistics is just because you have achieved statistical significance does not mean you have measured something biologically significant.
Right.
They're not necessarily the same thing.
Yes.
And many ecological problems are the effect size, if you've taken statistics before. So the size of the strength of the relationship you're looking for is immersed, included in so many other contrasting and potentially conflicting things that finding a statistically significant change is really challenging in ecological systems. So that this is an argument both for and against statistics.
Right, because if you find it, then it's probably something real.
A slight thing in ecology can often be really important because it's in the vast morass of the world, it's reared its head up and said, this might be real.
I'm here. Yeah, it's true. Statistics are a human construct, though, and remember that. Yeah. Good. And again, that's a philosophical thing.
Yeah. And then, if you want to get even more philosophical, you can talk to statisticians who think about probability versus statisticians who think about likelihood versus Bayes. And then...
yeah.
There's a whole careers there.
Whole careers. And here's another one. Evolutionarily, this is so interesting. Yeah. So if it is photoperiod, let's imagine that, and it totally is photoperiod, by the way. But if it is photoperiod, which is like totally not intuitive, right? Why? Why is it not temperature? What's going on there? How might that evolve? What might the explanation be? And we were going to answer some of those things, but we ran out of time.
We're going to talk about them on Monday.
We'll talk about them next time.
We'll shift some of them and we'll talk about the honesty of different signals.
Yay.
So...
take care.
Yeah, and remember to keep doing what you're supposed to be doing, everybody. Yeah. And encourage your families to as well. Some of your families are in areas that have changed zones and colors and whatever, and they're third waving, second waving. Too much of this going around. We got to shut her down so that we can get back together again. Good luck on your midterm. Yeah, and that too. We will be monitoring emails. If you have any kind of disaster, write us immediately if your internet times out or whatever. Okay? Oh, there's a fracking song? How do I not know? Take care. See you soon. And we'll be here. We'll be here. Yep. I am